Welcome to CyanoEXpress website. CyanoEXpress is a database for Cyanobacterium synechocystis sp. PCC 6803. It contains its standardized genome-level expression data. These data were freely obtained from microarrays and sequences deposited by labs worldwide into public repositories for public use. Hence, it contains two different types of expression data: those derived from microarray platforms and those derived from whole transcriptome sequencing. In this video, we show how to access and use these data. In essence, CyanoEXpress is the only web server that allows the user to compare multiple expression data generated using different microarray platforms. Links to the repositories from which the original data were mined can be accessed by clicking on the sample information link, which will open two tables. In the first table displayed on this page, a label is assigned to each contrast to show whether the perturbation measured with respect to the control is purely environmental or it has a genetic factor, in which case it's labelled as a mutant. Other valuable information presented here includes the data source, the microarray platform used, the links to the data and any corresponding publications. In the second table shown on this page, details on the data for RNA-seq are shown. Because of the different origins of the data presented in CyanoEXpress, they've been processed in a standardized manner to avoid problems related to the use of different data processing and data formats. To access the processed data and the exploratory tools, the user needs to click on Gene Expression on the navigation bar. Here you can explore different heat maps depending on whether your interest is to explore the full dataset under microarray all perturbations or only data generated using synechocystis wild-type strains under microarray environmental perturbations. Else, the RNA-seq data can be selected. Heat maps for specific functional categories, including cellular processes and energy metabolism, can also be accessed through icons shown in the lower panel. By clicking on Microarray Environmental Perturbations, we access the exploratory tools available on this website. For example, the heat maps that will allow us to investigate the influence of different environmental factors on gene expression, such as sulfate starvation, iron limitation, cold, and high-intensity light stress. On the left-hand side is a thumbnail image of the full heat map. This thumbnail image is used to navigate the heat map. On the main window, only a portion or a zoomed-in part of this thumbnail is presented. The zoomed-in image is updated whenever we click on different areas of the thumbnail image. The zoomed-in or magnified heat map is similar to a tabulated table, in which the columns represent the different environmental contrasts, and the rows show the expression level of each gene under the different conditions with respect to the control conditions. Green indicates a lower expression of that gene under that condition compared to the control, while red indicates a higher expression. Selecting any given row on this heat map will update the magnified image, providing a new map with the given gene now located on the top row, followed by 20 genes with the highest expression correlation to this gene. This process can be used to identify potential functional roles of proteins encoded by hypothetical or unknown genes. The yellow bar on the right-hand side indicates the degree of correlation between this gene and the gene selected in the top row. Selecting on any of these gene IDs will directly link the user to the entry in CyanoBase in a new window. Here we see the data for this particular gene from CyanoBase. On the top panel, the user can input any gene ID or gene symbol, for example SLL0247, and by pressing Submit, information will be displayed for the expression of that gene. This is created in a new tabulation with only that gene shown for all environmental perturbations. Alternatively, several gene identifiers can be introduced separated by spaces, allowing you to compare the expression profiles of various genes. The size of the magnified image and that of the thumbnail image can also be modified by selecting different percentages on the radar and zoom tags, and then pressing change on the right-hand side of these tabs. The three windows on the bottom panel labelled Gene, Sample and Fold and Log to Fold Change display the information for the given image. These values are updated when the cursor is repositioned over the heat map, as shown. This allows the user to identify specific data points on the heat map. In addition to exploring microarray data, CyanoEXpress also can be used to visualize and explore a set of RNA sequencing data. These can be accessed back through the Gene Expression tab by selecting the RNA-seq database. The expression levels of the RNA-seq data correspond to transcriptional units, meaning that the specific expression of a gene in a transcriptional unit might vary depending on post-transcriptional regulation. These data allow the user to explore the expression of non-coding genes not typically present in microarray data. For further information on CyanoEXpress, please refer to the publications shown on our homepage. All new versions of CyanoEXpress are shown and documented under News. There is also a help section available. Thank you for your attention. We hope that you enjoy using CyanoEXpress for your research.